Οπότε είναι αυτό που λέμε σε στα ελληνικά physician scientist και θα έλεγα ότι το focus της έρευνάς του είναι σε systems neuroscience. Συγκεκριμένα ασχολείται με τη μελέτη των δικτύων του θείου σε φυσιολογικές και παθολογικές καταστάσεις, κυρίως όσον αφορά το primary cortex. Θα μας μιλήσει για τις μελέτες που έχει κάνει με μεθόδους αρκετά προηγμένες που αφορούν imaging και ηλεκτροφυσιολογία. Και θα ήθελα στο τέλος να πω ένα μεγάλο ευχαριστώ στον Στέλιο για το training που έχει κάνει στους μεταπτυχιακούς μας φοιτητές μέσω διαφόρων προγραμμάτων. Ήταν από τους πρώτους κριτικά διάσπορα fellows στο Πανεπιστήμιο Κρήτης και έχει στενές συνεργασίες με διάφορα τμήματα, όπως είπα και πριν, με το τμήμα ιατρικής, με το ΙΤΕ, με την κυρία Κοντράσια, με την κυρία Παπακούλια, από το τμήμα υπολογιστών. Οπότε, Στέλιο, ευχαριστούμε που έκανες ένα διάλειμμα από τις διακοπές σου για να μας μιλήσεις.
Εγώ ευχαριστώ, Domna, ευχαριστώ όλους τους φίλους εδώ που μου δώσαν την ευκαιρία να μιλήσω. Ευχαριστώ και τα παιδιά που μας ακούσαν στο μάθημα των βιολογικών δικτύων πριν από το φθινοπορεινό εξάμηνο. Και σήμερα θα μιλήσω για applications βασικά και στα αγγλικά, διότι αλλιώς θα κομπάσω και δεν είναι ωραίο. So I will speak in English. So my talk today will focus on applications of calcium to photon imaging on studying neurological models of diseases, specifically epilepsy. And as Domna said, our interest is in circuit analysis, so what actually is going on in the cortex that allows the cortex to engage into these epileptic patterns of activity. Now, of course, not all epilepsy syndromes are equivalent to each other, so we have to choose, and the first model that we looked at is called the Stargazer epilepsy, and it's called the Stargazer because, as you will see, the mouse kind of, when it gets a seizure, it looks at the star. It's an absent seizure, so the mouse has a behavioral arrest and looks up, so they called it a Stargazer. Jeff Nobels called it a Stargazer that described this mutation. I should say this is work that involves several collaborators, including Jokenmer is my postdoc that is most of the experimental work in collaboration with Atul Maheshwari, who was also Jeff Nobel's postdoc. This is a collaborative work also between us and Jeff Nobels who described the Stargazer mutation, and Shalom Jan, who is the preeminent patch clamper in the world. This guy is amazing. He can patch clamp about 12 neurons simultaneously and do circuit analysis this way. I really don't have any disclosures relevant to this. So to start, let me just describe absence epilepsy, which is a common human childhood disorder featuring frequent corticothalamic spike wave seizures associated with behavioral arrest. And what that means is that there is an abnormal wave of activity that propagates back and forth between the thalamus and the cortex, and the behavioral result in the animal is that the animal stops behaving during this. So to give you an example, you could be talking to somebody and then they freeze and then continue their discussion of where they left off after they froze for a few seconds and have no memory that there was a conversation in between that other people were communicating with them and so on. The stereotype bldg electroencephalogram signature of the absence seizure is often assumed to reflect global hypersynchrony. So the thought is that you get an increased level of synchrony among neurons across all cortical layers, and this, however, has never been exactly demonstrated in vivo in the cortex, and it's part of what we wanted to do here, to demonstrate the system neuroscience signature of the absence seizure at the cortical circuit level. The mouse, here is a video that kind of comes out. So you can see the mouse. These are the electroencephalograms, and you can see when the seizure happens, the mouse, just pay attention to when the bar reaches right before the seizure, the mouse will stop moving, will freeze. You may imagine it looking up a little bit, and then the seizure ends, and the mouse resumes its foraging. So this is the behavioral phenotype of this event. Let me just go to this again. The seizure is about to start, right? And the mouse freezes, and then the seizure ends, and the mouse resumes its behavior. So we don't quite understand what goes on for this to happen. But we do understand something about the molecular biology of the disorder. In particular, we know that this disease or this mutation that causes absence seizures in mice is caused by a mutation in the Stargazer gene, which really is a calcium channel, the carbon G2 channel, which is a subunit of the AMPA receptor complex, a TARP subunit, which is involved in trafficking the AMPA receptors from the membrane to inside the cell. So basically what happens when this is mutated is that the trafficking isn't as it should be, and there is fewer AMPA receptors specifically in the synapses between the pyramidal neurons and the parvalbumin interneurons. Stargazer, the gene, happens to be very close to the parvalbumin gene actually, extremely close, and therefore it's expressed primarily or almost exclusively in parvalbumin interneurons. So it affects the connection between pyramidal cells to parvalbumin interneurons, and this is work from Atul Maheshwari and Jeff Nobles, which demonstrated immunohistochemically by staining a subunit of the AMPA receptors that in the wild type you get more AMPA receptors in these synapses compared to the Stargazer homozygous. The end result of this is supposed to be that the parvalbumin interneuron cannot be engaged when you have activation of the pyramidal cells, and therefore it cannot inhibit the network if it runs into aberrant activity. So our goal in studying these disorders is to sort of move from the cell phase to the network phase. That's not to downplay the sequence of disease, which starts from the description of the disorder, then the gene that causes the disorder, then the cellular dysfunction that underlies the abnormality that reflects the gene defect, and then the network malfunction, which tends to be much more diverse than one might have expected from just a single gene mutation. So one of the points I'm going to try to make here is that even this disorder, which is a monogene epilepsy disorder, what we'll end up finding is multiple circuit abnormalities, which if you want to correct, you need to know a lot more, I think, about the circuit function than you would have anticipated. Anyway, so this last part is where we focus today, and I will describe what kind of malfunction this gene mutation causes at the network level. Interesting questions abound, but some of them are which cells are affected in the neocortex. There are multiple different cell types, and how is the network function altered. So the general, based on the immunohistochemical results that I just described, which is that the AMPA receptors in the connection specifically between pyramidal neurons and parvalbumin interneurons is weak, what you expect is that the abnormality in the circuit behavior will go something like this. The connection, let's say there is some excitation in the pyramidal network, it's trying to engage the inhibitory interneurons that are going to suppress it. But this synapse is weak, it cannot do that. So the parvalbumin interneurons cannot inhibit the excitation, which then builds up and then reverberates to thalamic or cortical circuit and generates this abnormal wave of activation. That's the theory. And I don't mean to imply that's necessarily cortically generated by this. It could be happening in the thalamus. There's some debate going on in this domain, but the gist of it is that you have this aberrant excitation cannot be contained, generated on cortical activity. This is the theory, but it has not been tested in vivo, certainly not in the functioning cortical circuit in cellular resolution. So to do this, we are going to use the now old technique of in vivo two-photon microscopy. And just to remind you, I'm sure you know it, but just a brief slide as a reminder, what is two-photon imaging, why it is called two-photon imaging. Two-photon imaging is just microscopy, okay? It's smart microscopy. The only difference from ordinary microscopy, to be a little the only difference, or the main difference, is that in the fluorophores that you use. So you use an infrared laser which emits photons that get absorbed by the fluorophore. In order for the fluorophore to fluoresce, 2 photons need to be absorbed instead of 1. That's the difference. What the effect of that? So 2 photons get absorbed and the fluorophore emits 1 photon. The result of this is that you have a very sharp focus, because the moment you need a very high intensity, so the fluorescence that you get by having a beam scan your sample goes like the intensity of the impinging beam square, right? So the moment you move away from the focus, your intensity drops. So you can get very nice focused fluorescence from wherever you shine your laser beam. So the way you image the tissue then is you open, and this allows you to go, I should say, very deep into the tissue and to image actually the cells and the cellular activity in vivo by using appropriate dyes. So you can use a dye that is uptaken by the cells of the living mouse. You then go and open a window in the skull here, and you put a cover slip on top. And these windows, if you do them well, can last for months. And then you have, you put the animal under the photon microscope. The laser is shown, you shine the laser at the location that you want to image, and scan it. You move it around so you get a scanning image, and which you then reconstruct digitally on the screen. And you can image what you like, in this case the vessels or the cells or whatever actually has uptaken the dye that you activate with this image. So in this case we use calcium imaging, which means we use dyes like GCAM6 in particular, which fluoresce more the more calcium enters the cell. So therefore they can tell us about the cell activity, because when the cell spikes, calcium comes in, the dye fluoresces more, and the cell becomes brighter. And so it's by now the calcium signal is a well-established surrogate measure of electrical activity. In neurology here you can see a cell that has been patched, and on the bottom here you see actually the spike train from the patched neuron. On the top you see the EEG, and the neuron itself, the brightness of the neuron reflects the firing, the spikes. So you can do this, you pretty much can get a very good impression of the average firing activity. You may miss, depending on the dye you use and the sparsity of the firing of the cell, you may miss some spikes, but it's a pretty good measure of what the neuron is doing. This you can see by this plot down here, where by blue, this is just spontaneous activity, no stimulation. The blue curve corresponds to the electrophysiological number of spikes per second kind of firing rate, and the red represents the calcium signal. So you can see that they match fairly well, so that in this case, particularly layer 2-3, where cells fire sparsely, the calcium signal actually can get almost all spikes, and does pretty well for monitoring cortical activity. This is what the window might look like right after it gets, so the bubbles are still here on the surface, but the window you can see fairly clear right after it got open, and then it can last for weeks to months and allows you to observe the cellular activity. This is what a stack through the cortex, through such a window looks like, and you're going to see the cells here, and you can see how they flash as you are going down also here. So two things are happening. Some of the cells flash as they have the activity, but also your field of view is going down, and so you can see these cells, you can find them in repeated imaging, and you can monitor their properties. So the experiment is really very simple, and the idea is, well, okay, we have the EEG, which is the surface recording of the potential that appears on the surface of the skull when the animal seizes, and that is highly oscillatory in these mice. So you can have 6 to 8 hertz oscillation that typically lasts for several seconds. So this is a seizure, okay, that these mice have. And the question is, okay, how well are these neurons engaged into these seizure patterns in this absence model? So I would concentrate first on that. It turns out, and one of the lessons here is that they are not. So I would have thought, even for the absence seizure, which is not as entraining the cell as the grand mouse, I still would have thought that what we would see is that the cells actually lock onto the EEG very reliably, or mostly reliably, but that's not what we saw. So it's actually immense variability, and I picked up a couple of cells here to demonstrate the two kinds of cells there are, and I'll go more into the types of behavior that you see. But so you can see already here that as I move along, the seizure happens, let's say here, but no particular huge wave of activity happens. Now, if you focus on the blue arrow, you actually see that that cell fires mostly when the seizure is either over or before, so it doesn't really fires more in between seizure periods than in the seizure, and the red actually turns out to do the opposite. It fires a bit more during the seizure than before. So you can, at first pass, classify the neurons as ictal low or ictal high, ictal low being during the seizure they fire low. The activity gets suppressed actually. And ictal high, during seizures the suppressed activity goes up. You can get a quick view of what's happening on average across many cells, as well as their inputs that are in this area, by looking at the neuropil. The neuropil is the part of the axons and dendrites that are around the neurons, and they also uptake calcium, and you can also monitor that calcium. And you can see here what happens on average in a field of view of one of these animals when the seizure happens. You can see that this being the baseline of activity at the neuropil, and then right before the seizure starts, and actually several seconds, this is about several seconds before the seizure starts actually, the activity gets suppressed, it bottoms out during the seizure, and then when the seizure stops, activity goes up. That's the aggregate activity. That's actually the behavior during absence. It's a suppressive behavior rather than excitatory. And then you can see here the two different types of neurons that you generally see. This is the ictal low neuron, so follows the neuropil activity, its activity. Now it's a single cell here. It goes down with the onset, up with the offset. But there are also a few ictal high neurons whose activity behaves differently. It ramps up when the seizure starts and then goes down after the offset. So how do the neurons break down? I just told you that the majority of them appear to be suppressed, and this pans out if you look at the neurons. About 80% of the neurons recorded in layer 2-3 actually are suppressed, so they are ictal low. Only about 5% or so are ictal high, and then there are some that are neutral, meaning we can decide they don't seem to be preferentially firing during seizure versus in between seizures. And neuropil is 100% ictal low, so if you select patch simply because it reflects the aggregate. This is reinforced by the plots below are simply animal by animal or field of view by field of view, so you can see that this is not a fluke of the statistics, but every single animal has this tendency. Not only every single animal has this tendency by calcium, but if you patch the cells, so now we patch clamp in vivo the cells and look at them as the animal sees it, so you can ask, do the spiking activity of the cell is it higher interictally, in between seizures versus ictally, and you find that pretty much the same proportion holds. So out of, in this case, I displayed 9 neurons, 8 of them were ictal low, meaning they fired less during the seizure, and 1 of them was ictal high. And overall, we patched about 15 neurons and found 2 ictal high neurons, the red ones here, 1 of whom is barely increased the UV. So this agrees, generally speaking, with the calcium results. So now it's a little bit of a surprise, but perhaps not too much. Absence seizure seems to suppress activity in layer 2-3. Remember, the cortical layer 2-3 is the one that projects to higher areas. It's interesting to try to understand why this suppression ends up leading to behavioral arrest. But the first, you might want to understand what types of neurons. You have some ictal high neurons. Can you tell if these ictal high neurons are a specific type or not? Because of course one hypothesis is that the ictal high neurons could be inhibitory, right? They fire more and they suppress. Somebody has to suppress the cells, the majority of the cells which are pyramidal, which are firing less during seizures. So that's one question, do different classes of neurons behave differently? And the second question is what happens to the other layers? So we proceeded to check all layers, actually, and pretty much found the same behavior with minor differences. in the percentage of cells that are ictal low, but basically the majority, the large majority of the neurons are in all layers. As you can see here, layers labeled below, and the percentage of neurons, most of them are ictal low, and a few of them ictal high, perhaps a bit more related big layers, but not insignificant. And again, you can see this behavior in every animal, so it's robust. If you can plot out now how the activity drops as a function of onset of the seizure and offset of the seizure. The one I showed you before was one cell, but this is now the population of cells. So you get of the ictal low in this particular case. So just to get a sense of how does the EEG activity correspond to the actual, the EEG signature corresponds to the actual pattern of activity you see in the cortex. This is important in epilepsy because there is an idea of how do you predict. You need to have some idea, but it would be nice to be able to tell when seizure happens before it happens. The EEG onset of seizure is here. Okay. Here. But you can see already about 6 seconds or so before that, you can see the signature in the cortex that something is going off kilter. Yeah. Not necessarily the same experiment, but yes, meaning some of them, maybe 2 or 3 fields of view were recorded from the same experiment, but not all of them were. Not every experiment in others acquired cells from every layer, if that's what you were.
So basically, I'm going to measure for the data set, even if it's small in terms of space, of the same time period across layers.
Not. These were not done in a way in which the layers were interleaved. You mean wanting to have simultaneous acquisitions across it. Not here. We are doing these kinds of things, but with a different imaging protocol, with a different device actually, that allows near simultaneous imaging. These ones were simply, we selected the field of view in a layer, we imaged that, then we moved the field of view in a different location, we imaged that for a long enough time to be able to analyze the seizures that occur. So they are not simultaneously recorded. Yeah, I will talk about that. That's actually an interesting question. You know? So let's say you identify the character of the neuron. Does it stay that way, or is the network more dynamic? The answer is they change a lot, actually. And I will show you some data on that. But first, you know, one of the interesting questions here is, which is what motivates us a little bit also, is the EEG, right? We see the patterns of the EEG. We don't actually know what these patterns correspond to in terms of activity of the cortical neurons or of the subcortical neuron. So the bigger question here is trying to do, one line of research that we are pursuing is trying to understand this, right? There is all sorts of patterns in the EEG. Some are seizures, some are epileptiform, spike and wave, whatever. You know, what does it mean? I mean, you see this, and you see this in different types of epilepsy, and they actually engage the cortex in different ways. So we'd like to understand a little more what is the mechanism of that and what are the patterns that you see. But in here, basically, you see the temporal engagement of these neurons, and the important thing is that it starts before actually the EEG seizure, so there is something to understand in terms of what preparatory activity in the network gives to the seizure. This is actually to answer your question about how persistently ictal low or ictal high these cells are. So we monitored over several days a sufficiently long period of activity to be able to characterize the cells as ictal high or ictal low. Predominantly ictal low, the neurons are predominantly ictal low, as I said, at all time points. But as you can see here, they change in character. So this guy here on day 1 is ictal low, and by the time you go to day 2 in this case, or day 1 second recording, which is probably about 8 hours later, it's ictal high. Day 4 it's neutral, it remains neutral. The others become again from ictal low, ictal high, and so on. So in fact, about 64% of all neurons, we reported over about 8 days, so about 64% of all neurons changed at least once in character over this total time recorded. Of course, ictal-low neurons, there are many more ictal-low neurons. They are much more likely to remain ictal-low than the ictal-high neurons. But the bottom line is that the lesson from this is that the engagement of the neurons, at least in this case it's layer 2-3, I should say. We didn't quite chronically follow the deeper layers, but at least in layer 2-3, the engagement of the neurons to the seizure is quite dynamic. It doesn't always happen. Sometimes it happens, sometimes it doesn't. We don't understand what guides this. This is what happens when they do engage, right? So you can do this. The way to do this is to do patch clamping and look. I mean, you have the EEG up here, and you can see the spikes the EEG has, and below is actually a patched neuron. A patched neuron that in this particular seizure, not in all seizures this behavior happens, but in this particular seizure, it actually engages. And when it engages, what happens is it hyperpolarizes, and then the oscillatory activity happens. And this oscillatory activity, the peaks of it match exactly the peaks of the EEG, and then there is a probability of firing that tends to happen at the peak. So this is a pretty good signature of engagement. However, a neuron can display this engagement for some seizures but not others, and different neurons can engage or not engage. The maximum engagement, in the sense that the maximum percent of seizures that displayed this kind of tight engagement in a neuron was about 80%, and that happened in one neuron. Most neurons engage more at the level of 20 to 50%. So 20 to 50% of the time that you see EEG, you get this precise pattern in the neuron, in a neuron that you're recording from. This is granular neurons. So neuronal engagement to seizures is flexible. So one interesting question, and again, I wish I had the answers for you, but in part this talk is designed to be on preliminary data and on kind of engaging, on presenting actually, what I like to present in part is the questions that are still unanswered rather than everything that's been answered. But so it's quite interesting. What is it that guides whether a neuron will participate in this seizure or the next? We looked at various things. We looked at consecutive seizures. There doesn't appear to be in that particular model of seizure a very high probability. In other words, if a neuron engages in seizure A, the probability that will engage in seizure B that follows A is almost the same, almost independent from the fact that we have a higher probability. One question that's interesting was what happens to synchrony, okay? The idea is that seizures are hypersynchronous states, okay? And certainly the EEG looks that way. You get oscillatory activity, and that means there is synchronization of subthreshold inputs. But again, at the level of spiking, this didn't pan out to be the case in this case. So if you look at the signal, actually, the calcium signal, or the deconvolved calcium signal, which I have in other figures that I can show you, the results are the same. These are the simple correlation, pairwise correlation matrices, and of the cells that were recorded. And here, this sector of the table are the ictal-high neurons correlated with the ictal-high neurons. Down here is the neuropil correlated with the neuropil. These two neuropil patches, many of them in the field of view. And you can see here that interictally there is a reasonable correlation matrix. You can see the level of the correlation very high for the neuropil, low for the neurons as expected. But ictally is clearly less. Okay? So the correlations drop, and they are significantly lower, both if you consider all neurons together or the ictal-low neurons. The ictal-high or neutral you cannot tell if they're significant, and the neuropil is also highly significant different. So there is the correlation, actually. So this was a bit of a surprise, right? So seizure is supposed to be a highly correlated event, but in this absence seizure model, there is the correlation. And this is actually true across all layers. So this, again, shows you the mean correlation coefficient pairwise in the field of view of each animal, and what happened with interictally versus ictally, so increase during the seizure, for the different layers, for the different animals. Okay, so somebody decorrelates the cortex, and then the next question is, this is mostly behavior that I described so far in the pyramidal neurons. Question is, how do the interneurons behave? One question is, how do we identify the interneurons? The prime interneuron we wanted to look at, as I mentioned to you earlier, was the parvalbumin one, because of this defect that's supposed to be between pyramidal neuron and parvalbumin interneurons, but of course we could not cross the line to the parvalbumin Crete because it had the gene is right next to it and it needed about 10,000 generations to make one recombination. So we had to use clarity, so we used clarity, so you know by now this technique takes the lipid off the brain and the brain becomes transparent, which allows you to image things in vitro. So what Atul mostly was did with Jorgen's help is to take the part of the brain that was imaged, process it with clarity, stain it for parvalbumin and somatostatin neurons, and then identify the parvalbumin neurons that we recorded from in vivo in vitro. So you match them so you know which one's stained. So in this case, you can see the alignment was done by looking at the it doesn't come out very well, but anyway, you can look at the blood vessels will help you align the sections. So these yellow arrows points to the blood vessels that help the alignment. The orange arrows point to the parvalbumin interneurons. You can see them here in vitro stained that were also GCAM6 positive in vivo, so we will get signal from them. And the blue here points to a parvalbumin interneuron that just didn't take GCAM6 and we couldn't say anything about that. But the bottom line is that doing this staining, we were able to identify parvalbumin interneurons as well as using some recombinations that didn't involve the parvalbumin Crete but picked up PLX mice, which have somatostatin and parvalbumin stain, and somatostatin Crete mice with a reporter that labels somatostatin neurons red. To make the long story short, it turned out, surprisingly to us, because we expected to find the interneurons to be ictal high, so they suppressed the cortex. But surprising, the majority of the interneurons were also ictal low. So both parvalbumin and somatostatin. All in all, in here, you can see that there were about 47 neurons, DLX, either somatostatin or parvalbumin. 4 were neutral, 43 ictal low. From somatostatin, there was only 1 ictal high, most of them ictal low, and parvalbumin that were identified via clarity, all ictal low. So the conclusion at this point is that cortical activity is suppressed in Stargazer mice, area B1, during absence seizures. Both neurons and neuropil behave similarly, and somatostatin and parvalbumin interneurons are also predominantly ictal low. This begs the question of who is suppressing the network, so it means that we don't understand something, and we'll go on a little bit further in now doing more precise network dissection. The coupling of neurons to seizure activity is dynamic on time scales of minutes to hours. Ictal lows are more stable than ictal highs, but neurons can change their character in long-term recordings. Surprisingly, pairwise synchrony between pyramidal neurons is also lower during seizures. There is a caveat here, there may be that at a very fine time scale some neurons are highly synchronized, but on average the majority of the pairs are desynchronized. Yeah.
About the low correlation.
Yes.
Correlation. But in the case that there is a line, so if you apply the visual correlation, then you miss this line, which has been important.
Yes. So in other words, you are referring to the STTC measure. Let's say some other measure. Yeah.
Or a different measure.
Yeah. So that's an interesting question, whether or not. So what Maria is saying is that the Pearson correlation is rather unforgiving, right? You would pick up things that are exactly aligned, and if you're misaligned by a little bit, you could still be correlated, but it would be missing correlated in the sense that you fire close proximity with increased chance. There are better measures of correlation. I think, as she alluded to, the best in my knowledge is something called STTC, which is a tiling coefficient, which is really very good at it, because you can define your own window and you can basically ask yourself, if you have 2 neurons fired, okay, you take one spike of one neuron and you ask yourself, what's the probability that there is a spike of the other neuron within a certain window from that spike, and what's the node distribution of that, assuming they are independent. This is much better than Pearson for allowing you to focus at the epochs where something happens. You know, the neurons fire versus the Pearson, which takes also the long stretches of nothing. We didn't do this in the long run. We didn't do this, and it's something that's interesting to do. As we have discussed during this issue, it's quite interesting to do a more sophisticated network analysis here. It's a different talk, but in normal cortex, we have some way of identifying groups of neurons that seem to be coordinating with each other to do certain computations. It's interesting to ask the same question with the same techniques here and ask whether there is groups of neurons that are coordinated somehow in eliciting this abnormal activity, but we haven't done that. Okay. It's important to say from the perspective of proper experimental controls that these things that I'm telling you about are not due to increased interictal locomotion. The reason this is important is because it's well known if the mouse is moving, the activity goes up in their brain. And of course, these are behavioral arrest seizures. So if during the seizure the mouse is behaviorally arrested, could this be just a byproduct of the arrest? But it isn't, because we selected periods to exclude the locomotion, and the same result happens. And it's not also the correlations, the increased correlations are also not due simply to the fact that there is increased firing in between seizures, because we control for that also. So now we're confused. I mean, the bottom line is that nothing is as simple as one might imagine, and this still underscores, I think, what is going to become a theme as we try to understand these circuit disorders. We need to understand a lot more than we suspect, okay, at the beginning. The main question is, can we understand the circuit pathophysiology? Can we actually understand why this is happening? And I don't have a clear answer for you, but I have some answers, some hypotheses, and this is a work in progress. To do this, in this particular case, we paired with Brigham, who is a preeminent patch guru that I talked to you about. For a classic paper by him is a Jangal 2015 Science, complete classic. Basically, is able to patch 8 to 12 neurons simultaneously and then pairwise stimulate them, so it gets the connectivity. The ground truth about the slice connectivity, it's not in vivo, it's in slice. And he's described all sorts of types of neurons in the mouse cortex, some of them known, some of them not exactly what people might think. But this is just, I'm showing you this to see the diversity of interneurons. This is just the interneurons in the cortex and what we are dealing with in trying to understand what the circuit is doing. So what he's doing is patching, as I said, multiple neurons, stimulating one, looking what the probability of connectivity is, and just plots, as he accumulates more pairs, plots a map, which I'm going to show you, a table, which has presynaptic and postsynaptic neuron, the probability of connectivity. So there are already some surprises here, you can see them down here. Remember what I told you that the main deficit is supposed to be here between the paravalvular, the pyramidal neuron, and the connection to the paravalvular interneuron. This is one of those connections there to 3 pyramidal cells, basket cells, the paravalvular, both the interneuron, and the result is there is no difference. So surprise number 1. Now, there is no difference there, but there is a difference in layer 5. So maybe layer 5 is dominant in here. But in other words, for layer 2, 3, there is not the same probability of connectivity, the same strength. For layer 5, as anticipated, the pyramidal cell to paravalvular interneuron is much weaker in the Stargazer. However, that's not the only change that you see. And perhaps more importantly, you see a connection between the basket cell, which is a paravalvular interneuron, and the Martinotti cell, which is much greater in the Stargazer. So the basket cell, which is a paravalvular positive interneuron, inhibits another interneuron of the Martinotti much stronger in the case of the Stargazer than in the case of the wild. And it's going to get confusing, but I'm going to try to paint the picture as I understand it at this moment. So bear with me. So this is the table I referred to a couple of minutes ago. What this plots is the probability of connectivity between 2 cell types. This is the presynaptic and the postsynaptic. So focus on this box for a second. This is the Martinotti cell. So Martinotti cell connects and inhibits pretty much all other neurons in the cortex, okay, including the interneurons. And this is the wild type. This is the strength of their connectivity. Notice here, the Stargazer almost gone. So Martinotti cell lost the capacity to inhibit the inhibitory interneurons as well as the pyramidal cells. That's one observation. Second observation is that the basket cell connection to the Martinotti, which, sorry, here is where I'm looking at, the basket cell, which is a parvalbumin neuron to the Martinotti, like I told you about. In the wild type, there is a little bit of inhibition, but in the Stargazer, there is a lot more. So again, this inactivates the Martinotti node. You know, in one case, there is no connection between the Martinotti, where there should be a strong one, and the neurons that it projects to. In the other, the inhibitor interneuron in the Stargazer inhibits the Martinotti excessively, whereas in the wild type it does not. So to make the summary of the deficits that we understand now in this, and they are preliminary. I mean, not all these boxes are filled yet. The gray ones are not actually checked. So it might be that this story becomes more complicated, but where we stand now is that there are two main effects in the circuit that the Stargazing mutation seems to induce. One is the suppressed Martinotti cell function that comes about three different ways. One is the pyramidal cell connection to the Martinotti is down, so it cannot activate the Martinotti. Second, the parvalbumin connection to Martinotti is up, so Martinotti gets inhibited. Third, the Martinotti itself connection to the other interneurons is way down, so the Martinotti itself, even if activated, is ineffective in activating the circuit. That's one problem. Now, of course, you can see how this can lead into suppression, right? Because if the Martinotti is not activating the interneurons, so it cannot inhibit the interneurons, then one of those interneurons that cannot be inhibited by Martinotti could be the culprit, could be suppressing the cortical activity. And there is a second phenomenon which seems to be less of a strong also, but less prevalent as we would have anticipated. This is what immunohistochemistry we have predicted here, is the suppressed layer 5 parvalbumin interneuron connection. So pyramidal, the parvalbumin interneuron is down, so the inhibitory interneurons of layer 5 are not as engageable. But of course, this should lead to increasing activity, which is not what we observe. Right? So that's probably a secondary phenomenon from the perspective of the circuit dynamics. So again, go back to how does it result in equal load, pyramidal, somatostatin, and parvalbumin interneurons? So we've shown that not only have we shown that the pyramidal neurons are suppressed, but also the two major control groups of interneurons, the somatostatin and parvalbumin, are down. So probably, as I alluded to, is a different type of interneuron that mediates this. And I suspect, although we haven't shown at this moment, is that probably the neuroagliform cells have played a primary role. And you can imagine this, Martinotti typically inhibits neuragliform. Neuragliform is a master regulator. It also inhibits everybody in the pyramidal. If you activate neuragliform, the cortex gets suppressed. So I suspect what is happening is the Martinotti not being able to do its job leads to overactivity of neuragliform, which then suppresses both the pyramidal cells and the inhibitory interneurons. Alternatively, there is another possibility, which might be that simply you get lower feed-forward drive from the thalamus, let's say, that activates the cortex, and this you can check by injecting stuff in the thalamus and looking at what happens when the fibers arrive in the cortex. But again, then you just transpose the problem of who is decreasing the activity from the cortex to the thalamus. So my suspicion is, although we check, this probably the top one is my favorite hypothesis. better than I did when we started this experiment, but it's quite clear to me now that especially for neurological disorders, where you may have a single gene deficit, but then you undergo through development, a lot of things change in the cortical circuits. So if we want to repair the cortical circuits and really do a good job at it, we need to, one, understand it much better, and two, develop agents that would allow us to selectively activate or inhibit particular nodes in the circuit. And this means at some point going beyond the simple, not so simple, but anyway, receptor-based pharmacology to cell-type specific pharmacology. This is much harder, of course. Achilles knows well that this is not trivial, but it is something that I think will come to pass at some point. And when it comes to pass, the degrees of freedom that you have to control the circuit will immensely increase. The circuits are controlled by neurons. They are not necessarily controlled just by, I'm sorry, I don't mean to make a too strong a point here. I think the receptor pharmacology is very important, right? And you can modulate receptors and adjust the circuit too. But if you just want to point out that if you had specificity at the cellular level, you could do even more. You could reset the set point of the circuit to increase plasticity, you could reset to stop seizures, and so on and so forth. So the conclusions, you already talked about what passed through this. But the gist of it is that an intensive, focused, and interdisciplinary approach is required to dissect circuit function in a well-defined epilepsy model in order to understand circuit malfunction and to identify appropriate targets for therapy. And I should leave this by saying that this is work of several people and I cannot praise them enough, particularly my postdoc, Meyer and Atuk, whom I supervised, and Jeff Roberts, who provided the mice and supervised Atuk as well. Anyway, and Sherlock is a force of nature. So thank you very much. Feel free to ask questions. Let's get this started. Yes, so good question. I will tell you that we did look, we haven't published, but we are in the process of looking in other areas, particularly the somatosensory. There was a reason we looked at V1. We did not want to look, we wanted to look at the so-called penumbra, seizure penumbra. The thought about the seizures in these animals is there might be a cortical focus in the somatosensory area. We really didn't want to look at that. The behavioral, because we wanted to study as a second level the behavioral arrest. So we wanted to look at an area which is involved in the arrest, but isn't necessarily the primary focus. It turns out that if we look with the calcium at other visual areas, as best we can tell at this moment on a preliminary level, the same suppression happens in the correlation. The somatosensory is where we need to look more carefully because there is a little bit of a mismatch. In other words, you look with the calcium, you see the correlation, but there is some waves of activity electrophysiologically that we don't quite see in the calcium, just specifically in the somatosensory area. So we need to confirm those and try to understand a little bit better with the patient whether there's something funny going on. But the interest we have now in looking at other areas is in V1 in particular is that we also did visual stimulation. So we really like to understand why the visual stimuli, what level the visual stimuli do not rise to perception, what is wrong. Right? The results are preliminary, but it does appear that things like tuning functions and so on are degraded during the seizure versus interictally. That's an interesting question to ask, how that is degradation, does it occur in V1 primarily or in higher areas and how it propagates.
degradation is only during the ictal period, or is it more?
The what, I'm sorry?
The degradation of the tuning curves.
Right. Well, as best we can tell preliminarily, we compare at the moment, because the problem is, so there are tuning functions interictally. Whether there is a subtle difference between those tuning functions and the tuning functions of control animals that are litter mates that do not have the mutation, we haven't checked in excruciating detail yet. What we have checked preliminarily, and even that's preliminary, right, is between, in the same animal, interictal and ictal. And certainly ictal periods degrade, and you might expect that, but I think we need to understand exactly how and where.
And one more technical, I guess, thing. In your slide where you showed the clamping with the EEG recording,
yes,
where you showed the oscillations, to me it looked like that the sub-threshold oscillations are actually gone. And you might have this maybe synaptic, I don't know what they are.
Where, which slide do you refer to? There?
Here, yes.
Yeah.
So if you look at the noise before the ictal period, before the, here you have noise, and all of a sudden your lines become very straight.
Right. So this is an interesting phenomenon actually. So this is what I refer to as this oscillation, right? This is a little bit of a hyperpolarization, and then you're right, things appear to go into this oscillatory mode. Okay? This phenomenon is actually, I didn't say it, but it happens also in normal mice. Okay? Quite interestingly, this oscillation, I don't mean the seizure. The EEG is not necessarily abnormal, but if you look at this pattern, with some differences perhaps in terms of duration and so on, but you see this pattern also in normal mice. And when do you see it? You see it particularly when there is some sort of behavioral arrest, meaning the mouse runs, for example, the treadmill, the mouse stops, you might see this. Okay? Things like this. So it's a bit suspicious. It's a real physiological phenomenon, okay, which presumably gets hijacked by whatever the stargazing is doing. So now it hijacks this normal oscillatory thing and causes and presumably recruits many more cells to meet than ordinarily would be the case.
Are you saying that this is an amplified due to the change of the state?
Yeah. So like behaviorally, it just happens if you record the mouse and the mouse is engaged into something and then stops this engagement. That tends to be when this pattern shows up, which is intriguing because in the Stargazer it shows up when there is behavioral arrest, which is kind of a similar thing in principle anyway to what the normal behavioral arrest might be. The mouse is engaged in looking at a particular stimulus to change the stimulus. It's a behavioral change. So in periods where there is behavioral change, it's more likely to see these events. I mean, we're not the only ones that see this. Peyman Bolshani has observed this, Andreas has observed this. This is a real phenomenon, a real pattern of activation. And again, we don't know at this moment how exactly it's generated and what its behavioral meaning is, but other than what we have.
If this is the kind of like refreshing or initialization of the state
Something, yeah, maybe. It might be. You might make that argument. But the mouse, can react during this period. So in the case of the normal mouse, the mouse, there's a behavioral somehow stopping doing whatever it's doing, but it's not ceasing. It's not the Stargazer mouse that undergoes this oscillation is arrested. It doesn't respond to external stimuli. The normal mouse will show this pattern in some cells that you have touched, but it doesn't arrest. Even if you stimulate it, it will react.
Thank you. Questions?
Got that?
So I can speak loudly. So you mentioned that the neurobiofilms are global happening.
Can be, yeah.
Have you demonstrated that under epilepsy, or have you seen it in the normal situation? And do you know
what the targets are?
No, the neuragliform have been mapped by Jan. Where are these slices? I'm not sure if that map has them. Let me go back. So if you look, this is the presynaptic neuragliform cells, and this is the connections it has with all these cells. This is the layer 5 neuragliform. This is the layer 2-3 neuragliform, and there is layer 1 neuragliform, right? So you can see that these cells actually connect to everybody, okay, and they inhibit everybody. They are really master regulators. You activate them, they shut down. Now, the layer 2-3 ones connect stronger to the layer 2-3 cells than to layer 5, but actually interestingly connect also to layer 5 with sufficient strength. And the layer 5 neuragliform connecting stronger to the layer 5 other cells. But this is, I think, well established, I think, from these experiments and others, that if you activated them, they would suppress.
They target the dendrites, right?
Sorry?
They target the dendrites.
The layer 1 at least, they can, yes. They would target the apical dendrite. The layer 1s. I don't know about the layer 2-3 or layer 5. Do you?
Very exciting. I need lots of time discussion with you in Boston to let you know what things do exactly. So I'm wondering if the use of different various anti-seizure or anti-epileptic drugs will help you dissociate the difference in reactions and responses to different symptoms.
Yes. They would, but the problem, of course, is that they tend to be not very specific, right? So there are better ways. I mean, we are engaged into doing this. So one is, of course, optogenetics, right? So at least experimentally, we have the tool. We simply have to control the different cell types. Still, the possibilities of what you need to control and what you need to read are a bit too many. So we preferred initially to go through the approach of do extensive circuit analysis and then try to see. It kind of becomes like the pharmacology, node maps, all sorts of things. I mean, you have pathways, right? And you have nodes in the pathways. Exactly the same problem comes time and again, right? This is the circuit, same problem. Pathways, nodes in the pathways. What do you control? What will have effects without having side effects? It's not so trivial. But I think it's the beginning, and I think it's becoming much more exciting. So from the perspective of experimental control, the optogenetics is the better approach. From the perspective of treatment, eventually, the optogenetics is difficult to deliver. Pharmacologically generating agents that can be deliverable much better.
So is there any similarity between this and migraines?
Migraines, yes.
Yeah, because that's
a
similar
depression. Okay, spreading depression. Interestingly, there is some relation, okay, but what exactly it is, again, we don't know. Spreading depression is a different, you can see spreading depression with a calcium imaging. I have somewhere an image of this. I can show it to you later because I think I'll need to pull up the movie. It turns out that these mice, we actually spontaneously, apart from the seizures, if you observe them long enough, you see these spreading depression events. Where they're spread, or at least you suspect that they're spreading depression because they propagate at the rate of spreading depression to the cortex. They are quite impressive. You see them as a massive. What you see is you somehow see at one point inhibition rise. Okay, so everybody gets silenced, and then they release from inhibition, and you get this wave of propagating activity. This happens. We can trigger these in normals with stimulation, actually. These waves. So yes. It turns out that in these mice who have more frequent spreading depression, whether that means they have migraines, I don't know. The other disease that they are related to is autism, actually, because actually the defects of this gene, the Stargazer gene, in humans are associated not with seizures but with autism. Even though the absence seizure that they get is a typical absence seizure phenotypic. The particular gene in humans causes autism. So the genes that cause epilepsy, autism, migraines, they're associated with each other. And again, something happens in the circuit that we don't understand. But I mean, one would think, you know, really, I think it's kind of at the stage of where the genome project was, right? People were arguing about the genome project when it was coming out. Would it be useful, would it not be useful? Here we have two choices. Either we're going to do, at some point, detailed circuit analysis in several cases and try to understand the circuits in detail, or we're going to be going specifically after what we think is the best guess. And even though, and this is why I gave you this talk, because here where the best guess was very clear from immunohistochemically, genetically, and whatever, was just not the right answer. So I'm suspecting that this is you're going to have to be lucky, you know, from my perspective. You really, if you choose put your eggs in one basket in the typical NIH or very hypothesis-driven fashion and try to do systems neuroscience, you're taking a risk. You know, I'm not saying it's always bad, but I think one should have in parallel develop ways in which one does the group analysis in some fashion.
But I think it also has to do with time, right? If you study this circuit very early on in the development of the muscle, it will be very different. So if you can intervene at the beginning with a very targeted drug, then you don't have to solve this puzzle.
Maybe, but the problem, that's correct. And so in a way, if you manage to correct the genetic defect before the development, that's a great way to solve a lot of these problems. Again, a difficult thing, at least at this stage. Perhaps in the future you do prenatal diagnosis and pick up all the genes and you find out the mutations and you inject some sort of inhibitory RNA in utero or something. But okay, yeah, that's possible. I think we're far from that. I accept that's a good way. I think by and large, the most common problems we have now is genetic defects that unfortunately emerge as disorders after development, and then there is a lot of problems that you just often even reversing the gene won't cure. Sometimes it does. I'm not saying it's a bad way. Yeah? Georgie?
Thank you very much, Stelios. I was just wondering how variable is the duration of the signal stays?
Not hugely variable. In these mice, you know, they're very stereotypical, last from let's say 3 to 8 seconds or something like that. Rarely they get to be more like 10, 15 seconds long.
So do you have any thoughts on how the network is rescued in that state?
Well, thoughts, yes. Proof, not. You know, one thought is it exhausts whatever is driving the oscillation. So neurotransmitter exhaustion might be one way. Eventually, it might be actually, in this case, it's mostly suppression, right? So let's say the neuroglial cells get activated and suppress everybody, and eventually their activation level drops down because they don't get enough input anymore for whatever reason. But I don't know. I mean, I think you know, we do need to look at what cells are important for actually. In other types of seizure, it might be more likely. I suspect this is not going to be exhaustion, like I told you at the beginning. In other words, it might be that neuroglia from start being activated, but I don't think this is an issue of exhausting the neurotransmitter, because these seizures can happen very frequently next to each other on occasion, so there's no particular reason why you've exhausted something. In other words, you have 2 seizures 3 seconds long that are 2 seconds in between that are normal. It's hard to imagine that it's the exhaustion that caused the ending of the first seizure. It's a good question.
So what we see on this slide is very impressive because it's very widespread. And I'm wondering whether you think some homeostatic mechanisms might be there in this mouse.
Right. Yes, I think that's probably what happened. I think that it might be, I'm not denying the genetic defect. The mouse has the genetic defect, and probably the primary deficit early on in development was exactly this, must have driven somehow the circuit abnormalities that we see. So I think this is plasticity, homeostatic perhaps during development. So I think that's what's most likely the cause of this. Now, could be because of the deficit, or could be also because of the recurrent seizures. So it's not entirely clear whether the direct homeostatic effect was just the fact that you get the genetic defect.
And that was the question. Do you think the gene itself is involved in regulating homeostatic pathways?
I don't know is the answer. That's actually a big question in epilepsy, as you know probably better than I do. But in the absence, it's a bit difficult to argue about this because people have absence seizures for a very long time. And yes, there are maybe some associated cognitive defects, etc., but generally subtle. So it seems that these seizures are tolerated well, and in general, they disrupt people's behavior, but most people with absence, with the routine absence seizures, they can have hundreds of seizures a day, and anyway, on the order of 100, and still function normal. Except that they don't, they lose some parts of their. So I don't know that for absence seizures, the seizures themselves would play a tremendously strong effect in shaping the circuit. They might, I would be surprised, but I would need to demonstrate. In focal seizures, they certainly do. So in acquired focal seizures, the more you seize, the more you seize. And so to have a repetitive pattern of seizure in the brain is not a good thing. If you have a prolonged seizure, as you know, and you go into status, you then damage the cortex too, so not only are you more likely to get seizures, but you damage the brain and you lose neurons. So I think in this case, maybe not as strongly, but in the focal epilepsy, acquired